Okay. Waiting for my signal. Good evening. My name is Jill Traynor. I'm Dean of the College of Natural Sciences and Mathematics here at Sacramento State. And I'm pleased to welcome you tonight to our talk, which is presented by the College of Natural Sciences and Mathematics and the Sacramento State Center for STEM Excellence. STEM stands for Science, Technology, Engineering, and Math. The center works with area schools, colleges, universities, and employers to create a seamless STEM pipeline for scientific and technical workforce. This is the first presentation of the year in our series of public lectures on regenerative medicine. This academic year, Sacramento State will host 4 lectures by prominent scientists in the field of stem cell research. The lecture series is funded by the California Institute for Regenerative Medicine and supports our master's program in stem cell research. Earlier this fall, we were very excited to welcome our 3rd cohort of graduate students into this graduate program. Students in our 2nd cohort are currently conducting research with our partner in this program, the UC Davis Stem Cell Program. As part of disease teams, students are working with scientists and physicians on cures and treatments for disorders such as neurodegenerative disease, spinal cord and brain injury, epilepsy, HIV infection, osteoporosis, blood disorders, and burns. Sac State, and I'm proud to announce that Sac State graduated its first class of stem cell research professionals last May. It now gives me great pleasure to introduce this evening's speaker, Dr. Jianmin Zeng from the Buck Institute for Age Research in Novato, California. Dr. Zeng holds the titles of Associate Professor and Director of the CIRM Shared Research Laboratory of Stem Cell and Aging. She has a master's and a PhD degrees in molecular biology from the Technical University of Denmark. She directs numerous grants from the California Institute for Regenerative Medicine and the National Institutes of Health, and is author of over 40 papers on stem cell research. And I might also add that Dr. Zeng teaches a 5-day intensive course, a workshop for our students in our master's program. Our students have the privilege of spending an entire week at the Buck Institute studying with Dr. Zeng and her colleagues to learn about the theory and practical techniques of working with stem cells. And I hear wonderful things about this course, and I'd like to thank Dr. Zeng for all she does for our students. So please join me in welcoming Dr. Jianmin Zeng.
Well, thank you so much for the nice introduction. And thanks so much, Christine, Tom, and Tom for the invitation. It is really my pleasure to be here tonight to speak to you all. So I take it as an honor, and I have such a pleasure to teach the students from Sacramento State, the first 2 cohort of your stem cell program. I'm so happy that you come into my course and you think good about the course. And I'm so happy that you all found a job for the first graduate of you, so that speaks it for itself. So I think I'm just going to go direct to the topic of my talk today. So I'm going to talk about my work on neuron differentiation of pluripotent stem cells. So I'm going to focus on 2 topics, and these are the 2 most promising or potential, whatever you call it, the therapeutic applications of pluripotent stem cells. One is for cell replacement therapy, and the other is to use these cells for drug screening and hopefully find some of the drugs that can treat those devastating disease, including Parkinson's disease. I was told this audience is going to be a mixture audience, so it's not students only. So I think it will benefit the audience if I just give a few slides of introduction of stem cells. So what is a stem cell? You all heard about that. To define a stem cell, they need to have 2 properties. One is called self-renew. That means they can make more of the same cell type, okay? Like this. Second property is to have the ability to differentiate. That is to say, to give rise to 1 or more cell type in the body. In human, we have over 220 different cell types in our body. And depend on how many cell types a stem cell can give rise to, you can categorize stem cells into different categories. With the category called totipotent, this is the type of cells have or has the most potential to become any type of cells. Indeed, in human, these are the cells, the first zygote or the fertilized embryo, and the 2 to 3 cell stage. It can make an embryo plus extra embryo materials, for example, like the placenta. And then comes the pluripotent stem cell, which is the focus of my talk today. So pluripotent stem cells can be isolated from the very earliest stage of embryo development. And it can give rise to all the cell types, like I just mentioned, over 220, but it cannot make extra embryo material, like placenta. And then you also heard about adult stem cells. Those are mostly multipotent cells. They can give rise to several types of the cells, usually within the same organ, like you have bone marrow stem cells and other neuron stem cells and other type of the stem cells in the adult tissue. Okay. So as I said, I'm going to focus on pluripotent stem cells. When you think about pluripotent stem cells, there are two common sources for pluripotent stem cells. First is called embryonic stem cells. As I just mentioned before, you can isolate embryonic stem cells from the very earlier stage of human embryo development. In human, it's around 1 week after in vitro fertilization. So you can isolate embryonic stem cells from this called inner cell mass, and then you can culture them in vitro in a petri dish. And in theory, you can culture them indefinitely in culture. And these cells can become any type of the cells in your body. There is also a technology called somatic cell nuclear transfer. That is actually the technology that Dolly the sheep was cloned, okay? What you do is you take a somatic cells, for example, your skin cells, you take the genetic material, that's the DNA, you put it into an egg that you remove its own genetic material, and then you let the egg to hatch to an embryo, and you can isolate embryonic stem cells the same way as you do from an IVF embryo. So that is actually published 2 weeks ago by a group in New York Stem Cell Foundation. So the first time they showed this technology can be used in human cells too. So the third type or the second type of pluripotent stem cell is the so-called iPS cells. This technology is pioneered by the Japanese investigator Shinya Yamanaka in 2006. He used this pioneer technology to reprogram skin cells back into embryonic stem cell-like cells without going into the embryo stage. So this is something I'm going to talk a lot later in my talk. So just keep in mind, those are the typical pluripotent stem cell source. So the question comes, why are we interested in pluripotent stem cells? So I can give you many, many reasons, but as the title of my talk indicate, I'm going to focus on two things. That simply is because of its therapeutic application. First, you can use these cells to generate all type of the cells in your body, including, for example, like nerve cells that potentially will be useful for treatment of neurodegenerative disease and other type of the degenerative disease. And you can use the cells to do the drug screening and test your drug target. Now you have human cells in a petri dish to test the drug. That's something very useful for the pharmaceutical industry. Okay. So before I go to the specific of how we are, developing cell replacement therapy for Parkinson's disease, I thought I'd just give you this overview. This is what my lab is focusing. We try to make different cell types from pluripotent stem cells, and my lab is really focused on generating cells of the central nervous system and the peripheral nervous system. And for the central nervous system, we go through a stage called neuron stem cells. And these cells can make neurons, astrocytes, and oligodendrocytes. Those are the 3 cell types in the central nervous system, in your brain and in your spinal cord. So peripheral nervous system, my lab also working on generating neurocrystal stem cells that mostly my lab is interested in making Schwann cells. That is the cell type in the peripheral nervous system. I'm not going to go to the detail, but just give you an overview of this. So again, my talk today is going to focus only one type of the cells. That's the neurons, and more specifically, dopaminergic neurons of the substantia nigra type. So for many of you here, you of course know why people are interested in studying dopaminergic neurons. So one of the big reasons is the death of dopaminergic neurons in a specific part of the brain called substantia nigra. That's the cause of Parkinson's disease. When these neurons, dopaminergic neurons here in the substantia nigra, they projected to the striatum, and in the striatum, they regulate other neurons involved in motor coordination. So when this type of the cells die, so you lost the movement coordination, and the Parkinson's disease symptoms start developing once 80% of the cells are gone. So currently, there is certainly no cure for Parkinson's disease, although there are many treatments, including both pharmaceutical drugs and surgical procedures, like deep brain stimulation. All these are just to treat the symptom. They are not cure the disease. And as the disease progress and all these treatments, they affect kind of fade. And there is really a need to develop new therapeutic treatment or even a cure for Parkinson's disease. So that's when cell replacement therapy came into the picture. So that's the focus of my talk tonight, and at least the first part of my talk. So far, there have been 3 clinical trials have been running using not embryonic stem cell derived dopaminergic neurons, but the dopaminergic neurons derived from aborted fetus. There were 3 clinical trials has been done based on this replacement therapy. So although the outcome of this clinical trial are not very conclusive, some patients get better, some patients actually get worse. But there is at least a proof of concept that such therapy may work. So one of the big hurdles of fetal transplantation is the fact that you have a very limited cell source when you are using fetal cells. And the other issue is also you have no way to standardize the culture procedure when you use many of the fetals, and you cannot really expand the cells in a large amount. So for the clinical trial, they all use different procedures to prepare the fetal cells. So the outcome is difficult to interpret. Human embryonic stem cells has the potential to overcome this problem because, first of all, embryonic stem cells can proliferate indefinitely in culture, and it has the capacity to generate dopaminergic neurons. So that means, in theory, you have a source that can produce dopaminergic neurons in an unlimited amount that you can use for future clinical trial. So I'm going to, walk you through the strategy of our cell therapy development. So I listed here what I call there are different phases you're going to go through when you are thinking about develop a cell replacement therapy. And I put some of our collaborator and cell source here, just you have an idea. So I'm going to walk you through each of these steps, but just let me summarize in the beginning so you know where I'm going to talk about. So first of all, you need to select a line, an embryonic stem cell line or an iPS line that is clinic compliant, and they can make dopaminergic neurons. And after you identify such a line, you need to be able to generate a scalable process, not just making dopaminergic neurons in a Petri dish. You need to produce them in a larger amount. So this is very different from what you're doing in a research lab. So you need to have a procedure to optimize the differentiation process that you can make such cells in a larger amount. Once you have that, then you really need to do it in a GMP facility that is under the FDA's guideline for clinic use. And here you have a GMP facility in UC Davis. Many of you maybe perhaps seen it. So you need to manufacture cells in such a facility that can be used for preclinical studies and for safety tests for clinical trial. And finally, of course, we need to work with the clinicians to design a phase 1 trial. And we are preparing the material to file an IND with the FDA. So there are many of my collaborators and that I listed here, and I'm going to acknowledge it later also. So first step, that's what I say, you need to identify a clinic grade line. So in the past many years, we have worked with different biotech companies and other research institutes. We've been able to develop a media that is completely defined without using the feeder and scions and without using any animal original materials. And this is called xeno-free media. So we are able to grow embryonic stem cells in such a condition, and we have shown many of the lines that maintain the pluripotency and a normal karyotype that can be used for clinic applications. I also want to mention we are able to derive the lines, not only adapt the existing lines to this culture condition, we are also able to derive ESL lines from IVF embryo under completely defined conditions. So the next question is to ask, can we make dopaminergic neurons from such a clinic compliant lines? So in the past 3 years, we have published a serious paper to basically demonstrate this is possible. And we have done it with both embryonic stem cells and iPS cells. And these are just a few of the publications I listed here. And I'm going to walk you through the step how to make these neurons. I think many of the students are more interested in knowing the details. Again, I put a summarized slides here. You start with embryonic stem cells, and you you can also start with iPS cells. So you culture them in a defined media. You make neuron stem cells, and this is a procedure take about 2 weeks from undifferentiated cells to neuron stem cells. Once you make the neuron stem cells, we have develop a media and a culture procedure that we can freeze the cells down and thaw them again and again. Okay, so you can make many of them. And it does not look that simple like this diagram indicate. It takes many years of hard work, but we can do so. And the next step, we have a media that we know to induce dopaminergic differentiation, and we make it in a precursor stage. So that's the stage which we think will be used for future clinic trial to inject it into the patient. And finally, you can mature the neurons in either in vitro. For clinic trial, you will let this do in the patients or in vivo. So we have also developed a lot of assays technology to test whether these cells are going to be function or not. So first step, that is, I think this is always much better for you to see in pictures how the cells look like. Okay, so we start with embryonic stem cells, and we make embryonic bodies, and then you differentiate them into neuron stem cells. As you see here, the structure actually look very much like in the in vivo neural tube development. Okay, you isolate these neural tube-like structures, and grow them in a monolayer. That's the neuron stem cells. And we have a panel of markers to characterize them, that they are indeed neuron stem cells. And it's very important you do it in the right way, because that will affect your downstream differentiation. You need to have the right patterning to be able to make midbrain dopaminergic neurons. So what we do is we can generate a homogeneous layer of neuron stem cells that uniformly express neuron stem cell markers like SOX1, nesting here, PAC6, Musashi, or are neuron stem cell markers. So more importantly, we can differentiate these cells into neurons, express beta-3 tuberin, and we can also make a population of quite pure population of astrocyte, and we can make oligodendrocytes. And these cells can be cultured in vitro for more than like 20 passengers, still maintain the full capacity of generate into the 3 type of central nervous system cells, and they still maintain the expression of SOX1 and other neuron stem cell marker. For me, the most important question is whether we can make dopaminergic neurons from these neuron stem cells. So to answer the question, the short answer is yes. I'm going to show you a lot more evidence how the functional of dopaminergic neurons generated from this population. But this is just an immunostaining of tyrosine hydrolase, which is the rate limit enzyme in the synthesis of dopamine, which is used as a dopaminergic marker. So we can generate, depend on the differentiation period, you can make a large population of the neurons that express tyrosine hydrolase. And this is a picture take 5 weeks after differentiation. We can get about 40% of the cells express tyrosine hydrolase. And this is out of the total cells, not only out of the neurons. So I'm going to show you a lot more of the characterization later in the essays, but let me just go to one of the related problems when you're thinking about clinic trial. So first of all, you want to make sure you want to remove undifferentiated cells in your final product, because this is a pluripotent stem cell-based cell therapy product. It's very important. Safety cannot be overplayed, so you have to be sure that you absolutely don't have any undifferentiated cells that theoretically can cause tumors, okay? You don't want that. And we have been working with a company called Dyna that is owned by Life Technology. They have been developing the beads for deplete undifferentiated cells, and we have tested their technology in our stem cell culture. I want to emphasize our culture has been in vitro for many passengers, even after the neural stem cell stage. So we never seen any pluripotent cells identified, for example, like immunostaining with OCT4 and other of the pluripotency marker. But still, we want to have a procedure that we can deplete undifferentiated cells. And use the same type of the bead. So I do want to mention the bead. This is one of the few beads that are approved by the FDA for clinic use. That's why I'm using it, okay? So for positive selection, as I mentioned to you before, we can get about 40% of the cells that are dopaminergic neurons, but do you need more pure cells? So there is always a question to ask. We want to make sure that if it's needed, then we can do so. So we have a bead sorting procedure that we again use the magnetic bead, the Dyna bead, use a neuroprogenitor marker called PSA-NCAM to isolate neuroprogenitor cells in our culture system that is pre-differentiated into dopaminergic fate. So with that procedure, we can get about 80% of the cells express tyrosine hydrolase. So when I'm talking about the manufacture, it's very important not only you can get the cells, but it's also very important to test them, and you need to develop assays how you're going to test them that are the right cell types for example, future clinic use. So I'm just going to talk a couple of things, and when I'm talking about the manufacture, it's very important you can break the differentiation stage into different stage. If you take it from undifferentiated embryonic stem cell to the terminal differentiated dopaminergic neurons, that's a long process. And for manufacture, it's not so easy. If you take many, many weeks, even months, and you manufacture them, and it's difficult to keep the process, everything going right. So what we try to do is to break it down into different stages. So you can store the cells at different stage, and then you can take it from there. You can characterize cells at the different stage, right? So what we have done here, as I told you before, we have developed a protocol that we can freeze the cell down and make sure that they are still maintain the capacities to differentiate into neurons, into dopaminergic neurons. And this is an example. We actually have manufactured the cells in a GMP facility, and my partner at City of Hope is doing this part of the work. So they expand the neurons themselves in the GMP facility and make sure that they can still differentiate into dopaminergic neurons, just like what we have done in our labs. So we take it a further step. Again, this is a picture, looks quite simple, but this is again takes us years of work to get it here. Once you get the cells to the dopamine progenitor cells, you freeze them down, you can freeze them down, but the problem is how you're going to store them without they dying. Okay, so this is a procedure we have been working very hard because, when you want to give it to the patients, you would like to be able to store the cells in a frozen vial. You can give it to the clinician. Instead of you give them a flask of cells, they don't know how to treat them. So this is important for manufacture. So we have developed a procedure to freeze the cells down at the stage where we think is going to be at the transplantation stage. So again, we need to make sure the cells can recover after the thawing, and they can differentiate into dopaminergic neurons just like the cells haven't gone through freeze and thawing cycle. So this is what we have done in the lab scales, and we are right now transfer this technology to the GMP facility to hope, of course, that it will give us the same result. And then the other thing I talk about the function assay. These are some of the assays we have developed. So for access the function of the cells you manufacture from a GMP facility, you do more or less the same as you do in a lab, but they need to be carefully accessed. Otherwise, you would not know if these cells are going to be useful or not. So we have developed a lot of antibody staining procedures to look at not only TH can also express in other type of the neurons or other type of the cells. So we also look at the midbrain marker and more specifically look at the markers in the substantia nigra. So these are just a few of the antibody stainings procedure. I'm showing it here. We have also developed a panel of markers used in quantitative PCR to look at both midbrain and dopaminergic markers. For study the function, of course, we routinely run, for example, like dopamine release test. That we want to make sure this dopaminergic neurons, they can release dopamine upon stimulation. And this is one of the example. If you stimulate the cells with a high concentration of potential, that they will release dopamine. And this is just a HPLC graph that show once you stimulate the cells with KCL, you see the production of dopamine. Without KCL, you do not see the production of dopamine. And we have run a lot of electrophysiology to look at the function of the dopaminergic neurons. Again, it's important to see the neurons, they have the action potential, and they can respond to different neurotransmitter, okay? So the last functional study, which is, I think, is the most important one, is to look at the function in vivo. So we use one of the well-accepted model for Parkinson's disease, and this is the 6-hydroxydopamine model, which you lesion one side of the striatum in the rat. And after the lesion, you wait for like 5 to 6 weeks until you know the dopaminergic neurons are gone on one side. And then you transplant your cells, in this case is human embryonic stem cell derived dopaminergic neurons, into the brain or into the one side of the striatum, and then you let the animals recover, and you do behavior studies to see if they can at least provide some behavior recover accessed by different parameters. And one of the example here is we use the amphetamine-induced rotation test. This is one of the tests to access the behavior recover. And as you see here, after 4 months of transplantation, the animals receive the graft. They get significant improve in terms of Parkinson's disease. And if you look at histologically, you look at the graph, you can see the donor cells that is labeled by human antigens that are in the graph and survive. And that's hopefully is what perform the function for the behavior recover. So this is most of the 3 phrases I'm talking about. So I'm going to just go through the last 2 phases, but I'm not going to go to the details of this, as most of this are going to be tested using the cells that are generated from the GMP manufacturer. I just want to walk you through what kind of the studies you need to do before you can correct all the data for IND application. So for animal studies, as you see, we have one of the most important things for phase 1 is really just to look at the safety, okay? For safety studies that we are working with the Jackson lab, that once we get the cells manufactured in a GMP facility, so they're going to look at the safety parameters, look all the things like biodistribution, dosing, and all these things you need to provide the data to the FDA in your IND package. And we also have another safety study because this is a surgical procedure, so there is a device involved. So we're working with UCSF. They have a device grant to look at a device that you are going to use to inject the cells into the brain. So this may use a large animals and maybe use the non-human primate or other large animals. Okay. And for efficacy, I just show you with the 6-hydroxydopamine models in the rat that you can see we can show at least that these cells are going to improve the Parkinson's disease in rat. So we also have a collaborator that are going to look in non-human primate model, but this is not required by the FDA for phase 1 study, but we have this in plan. So there are also other additional tests that you can trace the cells, and these are all, in our technology development. So, but these all are not necessary for a clinical trial, but it's something that I think is very important to look at. So for clinical trial, I'm not going to go to the details again. So I'm working with a different group of clinicians to assemble, at least all the data that you are going to be reviewed by the clinicians and develop a phase 1 trial protocols. So these are the downstream work, but in good planning at this moment. So just to summarize what I have told you so far, so I hope I have convinced you that we do have a protocol, that we know how to generate or adapt the existing cells for clinical use. So we can grow the cells in a completely defined media without they getting abnormal, okay? And we have a procedure that we can generate neuron stem cells under defined culture, and these cells can be generated, these cells can generate dopaminergic neurons again under defined culture. And we have a lot of assays to access the functions rapidly. And I have shown you the scale up in a GMP facility is feasible. So we also have appropriate animal models to look at both safety and efficacy. So with that, I want to go to the next part of my talk. So I told you about cell therapy development, but in my mind, these cells will be very powerful and useful for developing new drugs, not only for the Parkinson's disease, but also for other neurodegenerative disease, or if you can differentiate them into other type of the cells for other degenerative disease. So just a very short background. So why do we want to use these cells for drug screening? So you will ask what the pharmaceutical companies currently today, what are they doing when they test the drugs? So the most common methods or the assays pharmaceutical company today they either use, for example, tumor-derived human cells like HeLa cells, for example, or they use primary cells that mostly are from animals that you can make, for example, neurons from rat or mice embryo, but it's very small amount. So both of these cells, they have like disadvantage. Tumor-derived cells, they are abnormal. And if you think about neurons in the brain, they are post-mitotic. They are not going to divide anymore. And their physiology is certainly very different from something derived from tumor, okay? And also, if you use animal cells, even you can get the primary cells, animals are going to be different. And you know, we are different from mice, for example. So that's, if you have a large amount of primary cells from human, and these cells, like neurons, they are going to represent the physiology of the neurons in your brain. So that gives you a powerful tool to test the drug. So when you have the result from testing on such cells, it's likely going to be more precise compared to what you test on cancer cells and other cell types. So to do so, we have in the past years to develop a screening platform. So this is all published, so you can look at, if you are interested, you can, feel free to email me if you want to know more about this technology. What I want to say is just, here the published papers show we can do the high throughput screening. And in this case, you grow the cells in 96-well format, okay? And you can run a screening by robot to read. You have some assays. In this case, we use the ATP assay just to look at the cell death. You can do it automatically. That is important for screening, okay? And the second piece of the screening important for neuroprotection is, back when I was at the NIH, we have already developed an in vitro assay that we use dopaminergic neurons derived from human embryonic stem cells. We have shown that these cells are just like primary dopaminergic neurons in your brain, that they will be killed by a drug called MPTP. And they respond to gDNA, which is a neurotrophic factors. They will protect the dopaminergic neurons from MPTP-induced cell death. So this is already published. Then it took us many years to grow the cells in a 96-well format. And by this technology with MPTP or this in vitro model, we are able to run a small screen. This is just a small compound library with 500 drugs, and look at if there is anything that can protect dopaminergic neurons from MPTP-induced cell death. So all this to show you is just, this is feasible, okay? So then I want to put this picture here. Yeah, I think if you agree with me, this is very nice, right? So we have been developing the technology, and I've been working with a lot of biotech industries. One of the company I work close with is Lonza, one of the biggest biotech companies here. So they have estimate or evaluate our cells, dopaminergic neurons, and this is the neurons. We send it to them, they grow it, and then they evaluate, just make sure they can produce the cells in a larger amount. This is in a 96-well format again. And you can see the uniform expression of beta-3 tuberin, and a large amount of those cells are also express TH. So this is the screening platform that we are developing. Now I think I'm working with Lanza, try to, make this available to everyone. So with that screening background, and I really want to talk about is to induce pluripotent stem cells as a model for screening or to model the disease. So again, I talk a little bit about induced pluripotent stem cells in my introductory slides. So if you remember, I said this is a technology pioneered by Shinya Yamanaha in 2006. What he did is he just used the fibroblast or skin cells, and then he threw 4 transcription factors, and magically it turned back into embryonic stem cell-like cells. In the beginning, this sounds like impossible, how you can turn back the clock, like from adult back to embryo again. But it turned out it's true. Many other labs are able to reproduce the result, and we today do it routinely in our lab. So to the general audience, I think this perhaps will help you. What is IPA cells? So just think about IPA cells are like EA cells, except that they are personalized. You can get it from a specific patient or person. So if the person has a genetic disease or have other disease mutations, now you can make a line that carry these mutations, which will be very useful for disease modeling and test the drug. And this is something more. Now you can also compare the gene expression that may allow to make some discoveries that is not possible before you have such lines. And you can collect the mutations if it's a genetic defect that you can put it back to the patients. This is a combine of gene and cell therapy. And for transplantation, because these cells are coming from your own, your immune system will likely not reject them, okay? So I want to mention, before I go to the details of induced pluripotent stem cells, we have tested many of IPA's lines with the same procedure I just showed you before, making dopaminergic neurons, and we didn't find any significant difference. They make dopaminergic neurons just as well as your embryonic stem cells. And these are published, so I just want to show you one slide here. I didn't put an in vivo animal test there, but that also didn't make much difference. So I'm talking about the technology of IPAs and generation. As I said, Yamanaka pioneered the technology in 2006. At that time, he put 4 transcription factors delivered into the fibroblast using retrovirus. So the genes actually are going to be integrated into the chromosome. So that may cause some problems we don't even know now. So the newer approach for IPS generation, there are many new technologies. Every day you see a new paper, how people improve of this technology. So you can use less factors, and also you can use technologies that you now do not need to integrate your transgenes onto the chromosome. That will be very important for clinic use because you don't want to risk any of the mutation caused by this procedure, how it's going to affect the patients. So there are also other technologies like use small molecules and use RNA technologies, and then again, and direct the proteins into the cells. All these are new technologies. And I'm going to tell you 2 technologies my lab is using to make IPA cells. And I'm only interested at this point when I'm thinking about therapeutic application. I want to make the IPA cells without DNA integration. I don't want to get complicated, okay? So one of the technology we use is to use the episomal plasmid vectors, that you use it to reprogram the cells. Because they are episomal, they're not going to integrate into the chromosome. So as you grow the cells, they will be diluted out, Okay, so hopefully they won't affect your genetic materials. And the other technology we use, this is commercially available, use an RNA vector. So I'm going to go to a little bit details of each. So this is the episomal vector we use, and is generated by my collaborator at the Johns Hopkins University. So he make a vector that with all 5 reprogram factors under a common chromato control. And this is use the F11009 vectors. And he also make a lot of help vectors just to help the transfection being efficient, okay? So we have been using these vectors to generate a lot of IPS line. Before you jump into making IPS lines, I want to tell you this is a long draw, okay? It's easier to transfect the cells, and then you see something, oh my God, it looks like embryonic stem cells, and you thought you have a line, but that's not just so simple. So you need to go through a lot of characterization before you can identify a real line that is like behavior like embryonic stem cells. So for the technology, I told you this is normally it will take at least a half a year to get a line. So of course we have done this routinely, and we have done many of them. We have a lot of experience. Other labs are doing the same things. So just in general, I think this will be very good for someone want to make IPS lines. So just take this into consideration when you are making the lines. So I will start with the somatic cell type. You know, you can take a skin fibroblast cells, or you can also use other cells. I think still the best way to do is to use the peripheral blood. So that's the simplest way to get the cells. And also, peripheral blood, you can make CD34 positive cells, not that many, but it's possible. So easiest to do so is to take the cord blood CD34 positive cells. You only need about 100,000 of these cells to do the reprogramming. And even with the plasmid vectors, it's very efficient. So it's important when you do this transfection, you see the cells, if you look at the morphology, look like this. Actually, they look very much like embryonic stem cells. But until you run a live staining or pluripotency marker, you cannot really identify which one is the correct, which one is not. So I suggest that when you're doing the reprogramming, run a live staining around 2 weeks and pick them up. You can do this on the fluorescence scope, or you can mark them. So then you expand this single clone into a line. That will take many, many weeks. And you want to make sure they still are pluripotent. Sometimes they are so-called patisserie reprogrammed cells. After you grow them for a few passengers, then they just start differentiating. They lose the pluripotency. So that's important. After a few months, after you have passenger the cells, you check the pluripotency. And this is normally what we do after we have established a line. We look at the pluripotency marker, and this is just a few of them, for example, like OCT4 and NANOC, the positive expression. And the next step is you establish the line. Now you have banked some of the cells, and you still want to make sure they are pluripotent. These are additional of the markers. For me, of course, it's important to look at whether the IPA cells, they have the DNA integration or not. So after 10 passengers, they should be passed. And they should lose this episomal vector. So we check many of our clones in this case. You can see the positive controls are here, and with our clones, that they are gone. They don't have any of the vector materials there. So we also very careful to karyotype them. So for those who have not worked with iPS cells, and you need to be very cautious. So these cells in general are very easier to get abnormal compared to human embryonic stem cells. You need to select the right clones. So we routinely karyotype our cells every 5 to 10 passengers, and we do it often in the beginning. We don't want to take any risk that you spend half a year to work on a line, then you find out it's karyotype abnormal. It's happened very often from what I heard from my colleagues. So just keep that in mind. And the next thing you want to check is, are they really pluripotent? You at least want to make sure they can differentiate into the cells of the 3 germ layer. And the easiest to check is to go through the embryonic formation. And you look at the expression of the 3 germ layer markers. In this case, you look at the ectoderm. These are the neurons, and these are the smooth muscles, mesothelin cells, and these are the liver cells, and comprised in the endoderm cells. You can also inject these cells into SCID mice and look at the teratoma formation. That's a more powerful way to look at the pluripotency, but you perhaps don't want to do each of your lines, so depend on your purpose. But we routinely check the 3 germ layer differentiation. So to get to my research, so it's of course important. Can I make the specific neurons that I want? So again, we go through the same procedure. We adopt the cells to feeder-free defined media, and we make neuron stem cells, as you see here, and we can make dopaminergic neurons from there, and the cells also make astrocyte and oligodendrocyte. So now this is first the technology I talk about with episomal vector. So there is another technology if you want to do non-integration reprogramming. I would suggest you start with this. This is commercially available from Life Technology, and the method is much more efficient, and it's used RNA viral. The infection is much, much more efficient compared to the plasmid, and it comes with a kit. Not because I want to advertise for them, it's just simple. Okay, I have no interest to advertise for them, but from my experience at my lab, this is an easier way to do so. So now I want to show you a couple of the slides. This is the samples I have currently in my lab. So these are all collections from Parkinson's disease patients. Some of them with defined genetic mutations, and some of them are spontaneous or sporadic Parkinson's disease patients. So we have a collection of the fibroblasts from Parkinson's disease patients, and we currently generate IPS lines from all these Parkinson's disease patients, and I'm working with different collaborators to make sure that we have access to the same technology, making the same IPS lines, use the same method to differentiate them into dopaminergic neurons, and use this for drug test. That's the goal. So just want to show you one of the line we have generated. So this is a line generated from one of the patients here with a PARC2 mutation here, okay? So what we have generated is a line from this patient, and we are making neuron stem cells and dopaminergic neurons, and now we have many lines that we can compare also with the control and do more of the biologic study to test the drugs, okay, for the screening purpose. To summarize this, so I hope you know, just this is really just to give you an idea how you can use this type of the cells for your study if you want to do some of the drug discovery, or even just study the biology of the disease modeling. So I have shown you that we have a platform that we can do the automatic screening, use both the EACs and NACs. That is a paper we already published. So we can generate a large number of dopaminergic neurons from both EACs and iPSCs for screening. So we have an in vitro toxicity model for Parkinson's disease, and we are able to generate integration-free lines, and we can do so using Parkinson's disease patients' samples. And I'm not going to talk about the gene targeting. This is something else that is important for running the high throughput screening. You want to have other technologies that can read your endpoint, not only like a ATPSA. So if you have a genetic modified line with a GAP tag, for example, into TH, that you can read just by the GAP intensity. And there are a lot of imager softwares that can accelerate your research on that front. So with that, I want to show you 2 more slides before I let you go. Okay. So again, remember I showed you these slides in the beginning of my talk, so we can make many different types of the cells. And today I really focus on Parkinson's disease, on dopaminergic neurons. But the cells we made, the neuron stem cells, we can also make a very pure population of other cell types, for example, like astrocyte. And we also have a procedure, I didn't talk this part at all. We know how to make neurocrusted stem cells, and we have exactly the defined media, not the same media, but the same principle to make cells that will be likely useful in future clinical trial, for example, like Schwann cells for the peripheral nerve system regeneration. So I want to show you a couple of those pictures. These are the pictures or the staining of astrocyte, and you see this population, you basically don't see any neurons. Okay, we can make a very pure population of GFAP positive cells. This is after 5 weeks of differentiation. We can get over 60% of purity. And we actually can get more than 80% purity if we, let them differentiate for further longer term. And we also have a procedure to make Schwann cells, and this will be very useful for myelination studies. And actually, we just finished a study that we use these cells to do the myelination. And in vitro, actually, it myelinates neurons. So we're very excited about this other work. So I didn't show you all this part of the work, but I hope you can take the same procedure, think about what your topic is, how you want to develop therapy and to develop drug screening. So you can use some of the same principles. And with that, I want to thank my lab, and many of them work so hard to get you see the pictures here. And I want to thank my collaborator, Mahendra Rao is my long-term collaborator. He now is at the NIH Center for Regenerative Medicine. So we have worked for the past 10 years to develop all these technologies. And Larry Kucher is my collaborator at City of Hope, who run a huge GMP facility there. And Professor Lin Zhao Chen at Johns Hopkins, who is a pioneer of iPS technology. And thank you for your attention. Repeat the questions. I'm not sure everyone can hear it. So the gentleman asked a question: if you put the dopaminergic neurons into the diseased patient, will these neurons also get sick? Right. Okay. So that's a very good question. Actually, from fetal transplantation, one of the few postmodern, studies to look at the patients after transplantation, after patient disease for like 10 years later, they did find some of the neurons also accumulate, for example, like Lewy body, that is a symptom of Parkinson's disease. So that can happen. So Tom is curious to ask how mature or how determined these precursors are committed to dopaminergic fate. I cannot tell you exactly how committed they are. So what we try to do, this is just in our opinion, you have to transplant the cells at a stage that they are not fully differentiated, just because it's very difficult to keep them alive once you detach the cells from a petri dish, for example. But you want to push the cells toward this dopaminergic fate as much as you can. So you define the stage that you think is already, this is also mimic to the in vivo developmental biology. You have the patterning, you have sonic hedgehog, FGF8 there to push the cells to the ventral fate and to get into the dopaminergic neurons. So we have looked at the gene expression at different days, and we try to define a date that we think is pretty much determined to be make to dopaminergic neurons, but they are not mature. But this is also just our opinion. How much it will cost for a patient to get this procedure? I think this question I cannot really answer. I'll tell you what it will take for a clinic trial. So right now we have a grant from CERN that just to make a small size or a lot of about 50 vial of dopaminergic neurons, that should be in principle is be enough to run a phase 1 trial. That is, you're talking about 10 patients. And just to manufacture these cells, it cost us $6 million. So it's hard to say how much it will cost, but this is in very early stage of research work. So we need to get industry involved to be able to really put it into patients in the future.